I'm going to introduce you to Hans in a second, who's going to talk more about maybe what biotech is. And Hans is, when I started working in biotech, Hans was one of the first people I met, and he told me his story about he'd been touched by disease and how he had, I learned a little bit about Hans. And then he kind of, I can say that Hans was really a mentor for me in showing me the process, because it's all, at the end of the day, it's all about the process, right? And if we can make the process a little bit more efficient. And so I really started to understand the process of how things work through Hans's leadership and mentorship, and Hans is also playing now a big role in the foundation as a new member of our board. So it's my great pleasure to introduce my friend and my colleague and our board member, Hans Christinger.
First of all, it's really an honor and it's humbling to be here and speak with you. Does one can hear me? It's great to. That's later. And it's great to have time to meet you and learn about your families, and really just a privilege to be here. So thank you. First, so one of the first things when I was talking to Luke when he first joined us, he was talking about what his experience was when he and Sally had the diagnosis, and just the confusion and wanting to understand more about R&D and biotech companies. And so he asked me to talk about that a little bit today. So I'll kind of, it's a big topic. I'll kind of walk through a little bit of a story here. But, how many people are kind of comfortable with an understanding what biotech is, or companies that run clinical trials and things like that? So a few people, and okay, good. Well, I wanted to keep this kind of informal, and I have a few slides so that I don't ramble on forever and bore you. But don't hesitate to ask questions or jump up, or afterwards, don't hesitate to reach out to me. I think I'll tell you a little bit of a story about kind of how we look at this from a company perspective, from a biotech perspective. But really only two takeaways. You guys have gotten a lot of information, there's a lot to think about. So the two takeaways I would have for this is, one, there are companies out there that are incredibly passionate, and they exist to help and to deliver a treatment, to bring promise, and to do it urgently. So that's one thing. So there are companies with big resources that are there to help. The second thing is, as you go through and you have questions and you want to know more about what's going on or whatever it is, you've got a whole amazing networking team here to draw on. But count my name on that. So personally, don't hesitate to reach out to me. You already have Luke, so you've got everything you need, but you got another person on the team here for any time, really. So first I want to tell you kind of two things about me personally, so you have a little bit of an idea of who I am and who's talking to. And these are really transformational events in my life. And the first one is this picture. So I had been in the industry for a number of years. I spent the 90s at a company called Genentech, and I'll show you a picture of some of the work product there. Just beavering away in the lab, thinking we're curing the world and of all kinds of illnesses and things. And my mother came down with melanoma, and back then this was a death sentence. It's not so much today, which is great. But this picture was taken. She knew this was the last picture she'd have with her grandchildren. It was the last time she'd see them. And there's one grandchild missing because he was born a few months later. But so for me, this is really kind of impactful in going through that experience. And behind that story too, my father was in Becton Dickinson for a long time. So this was back in the day when insurance was different than it is today. There were no questions about insurance. My father-in-law, actually, a strange, funny thing is, my father-in-law used to work with Luke's dad back in the 70s and 80s. So a renowned oncologist. And so you think with this team that this would be something you could manage. And the thing that I learned was, it is awful. The system is just not working. It's very, very difficult. I learned how much you have to advocate for yourself, how much you have to educate yourself and be there, and take accountability for the things that are going on and the decisions about medication and trials and things like that. That was one thing that was a real surprise and eye-opener for me. The other thing was, I've been doing this profession for a long time, and I saw firsthand that treatments that we had looked at and passed on, because we didn't think they were scientific enough. We didn't think they were sexy enough. These were things that were critical. These were things that enabled my mother to take medication that she wouldn't have otherwise been able to take. So it really changed the lens of how I thought about what is important as we think about treatments and how we try to have an impact. So that was kind of one thing. And the second is this. Hope you don't mind me using it, Luke, but Luke, Sally, Susanna, Matt, this is really true inspiration. This meeting you guys has really transformed how I think about what we're doing, why and how. And the inspiration, the love, the support, the courage to go through difficult times to really find joy and love and happiness and working together on things that matter, figuring out what matters and work towards it relentlessly and urgently. That's kind of the second real kind of pillar in my journey. So now getting into it, I want to keep it very simple. First, what is biotechnology? I looked up, if you look up the dictionary, what biotechnology is, you get a definition that, honestly, I've got a couple of slides after this, but I don't know what this means. So the manipulation as through genetic engineering of living organisms or their components to produce useful, usually commercial, usually useful or usually commercial products. What does that mean exactly? And then there's a second part of it where you talk about various applications of biological science used in such manipulation. So I thought I would try to think about that and put down a couple of pictures for each one. So this is a structure of VEGF. So VEGF is vascular endothelial growth factor. This is a molecule, a growth factor that causes blood vessels to grow. You see it running rampant in oncology. You see it running rampant in macular degeneration or blindness. So we, back in the 90s, we solved this structure, and what you see on the ends are antibodies. So that's where the receptor normally binds and where it does its business. But I show this one first because these are protein things. So biotechnology usually think about expressing proteins and making proteins into medicine. So that's an example of the manipulation through genetic engineering, because we had bacterial cells. We put DNA into bacterial cells that grew up and purified it and turned it into a drug. So this represents Avastin, which is one of the most successful cancer drugs to date. And it also actually literally represents Lucentis, which is a big product for blindness. Both of those came out of Genentech. But that's one example of biotechnology, really innovation around science and medical application. The second example I thought might be a good example of the second part of the definition, which is various applications of biological science. So this is what we call a small molecule. And usually these are the types of things you think about from a pharmaceutical company, like the aspirin or the things that take as pills. So you don't think about it as biotechnology, but actually it is biotechnology. I think one point I didn't put on the slide is this is a derivative of the mushroom extract. It's just a ring that structures that's been formed versus what the mushroom expresses. But the biotechnological aspect of it are kind of the other 2 points. So with a compound this specific, people were able to learn a lot about what it binds to, what it sticks to in the brain. And so in the company that Luke and I work for, we're focused on rare neurological disorders. This drug is actually in phase 3 trials for Angelman syndrome. And through this specificity, we really understood kind of specific receptors that control your brain's ability to settle down and filter out signals and noise. But through the specificity of the chemistry, we could figure that out, which receptors you need to bind to and which ones you shouldn't bind to. And it's very different than any other drugs out there. So that's kind of one aspect of it. The other is a new understanding of what is meaningful change in syndromes and how to measure those in clinical trials. And I think that's really applicable for what we've been talking about these last couple of days too. So as part of this, getting to this phase 3 trial, our company ran the first industry-sponsored trial in this area, Angelman syndrome. And because it was the first trial, there were really nobody knew how to measure, what do you want to see in a clinical trial? What's meaningful change? How do you measure it? How much is enough? Nobody really had any idea. And the team did an heroic job, actually. So I don't want to go into the details, but the result of their efforts, now there's a new endpoint. So for the first time, the FDA has agreed to a particular kind of endpoint, which is going to be applicable for syndromes where you get a whole diverse range of phenotypes. So you've got a diverse range of symptoms due to genetics that are similar or different, but they were expressed differently in the children or in the patients. So that's one thing that, we're very confident will be successful, but, even if the drug doesn't make it, that's a huge thing for the FDA to take this step down. So, that's a regulatory change, but it's biotechnology. It's innovative, it's entrepreneurial, and it's coming from a company that's really using all the resources out there to try to drive some meaningful impact. So that's the second half of the definition. So hopefully the Webster definition makes a little more sense now. That's to me. So, and I just wanted to kind of bring back to, I think we're at a point where there's going to be a lot of change in kind of how we think about treatments and how we go about urgently developing those. So I want to kind of reflect back at, Genentech was started in 1976. And, the first products were things like growth hormone and insulin. And what they were doing was they were getting bacterial cells or CHO cells or different kind of cells to express these proteins. And then they would clean them up and they would be suitable for injection into children or into people. So where do you think the rule book was for that? When you go and you say, okay, what is the FDA going to let us do? There was no rule book, right? It was really about the community coming together. So clearly the scientists had to come together. They had to create the data to show why this was safe. They had to show why there was an hypothesis that was worth pursuing and how they looked at that in different models. But it was more than that. I mean, it was really the community coming together, the physicians, the regulatory agency would have to be willing to say, what? This is something meaningful. This is something we're going to try. There's some risks here, but we're going to do our best and we're going to try to move this forward. And I wanted to put that in there because I really feel like now we're at that time now. As Luke said, the science is there. There are a lot of understandings there for how to do this type of thing, and there's a lot of willingness from organizations like the FDA to partner. So it really is a community effort. So I wanted to also kind of highlight a little bit of how we think about, from a company perspective, how do we think about the development or the R&D? So there's some simple slides here, and thankfully they're consistent with the other slides that we've seen the experts talking about. But, as I said in the beginning, our purpose is really to improve care, to find care or cure, to find treatments that are meaningful, that are useful, that are helpful. And I've got a cycle here where it's really understanding the patient need, understanding what's going on with the kids or the patients. I'll go through the cycle, but you go through patient need, gene, cell model, animal model, and clinical testing. There's a cycle to it. There's a process to it, as Luke said. And if you look at it a little bit more closely, it's really important to understand the need. What is the child experiencing? And I put up here some things that, thinking about it from what we're working on, maybe some of these things are familiar too, but the developmental issues, movement, sleep, communication. But what I should have put up also is those positive attributes, the ones that you don't want to lose. Those are just as important, right? So I was talking to somebody earlier who was talking about his son's hearing and what was using. Right? These are things maybe if you're only focusing on the negative, you don't think that, we want to make sure we measure that, we don't lose that, right? So those kind of insights and details are incredibly important as well. And then you want to understand what is the cause, and we spent a little bit of time, we spent a lot of time talking about that. But understanding at the gene level, at the protein level, what's going on, and where are the things that you might want to test hypotheses for interfering or adding some help to the system or whatever it is. And I'll show you a little bit about what that could look like. And then when you understand what it is that's going on, or you have a hypothesis for what's going on, you want to replicate it. You need to show in models that you can replicate the system, you can test it, and you can show that what you're testing is safe and effective in the appropriate models before you scale up and go into people. So I just put very simply from a cell model or an animal model, can you replicate what your child is experiencing in some way? It's not going to be the same, obviously, but are there certain elements of it that you can test and measure in cells? Are there certain things you can test and measure in the animal models? And then as you go forward, you're going to go into the clinical trials, and this is where things like natural history and all the work to get there are incredibly important, so that you can really make sure you're measuring the right thing. If you're not measuring the right thing, you're not going to be successful in your clinical trials. You're not going to get statistically meaningful results. So that is really critical. I want to emphasize again, it's really a community. So I stole one of Luke's slides from our company and I doctored it a little bit. And I just want to emphasize the partnership aspect. So our company is Ovid. We have partnered with a couple of pharmaceutical companies and an academic institution. And these are where we've gotten our medicines from. The drugs that we are testing for different areas. So we did a lot of work to understand the science and find where we might be able to impact the science and address the need. And then we went out and we looked for mechanisms of actions of drugs that fit that model. And these are two that, those are two that, those are three actually that we've gotten. We've got a head start, if you will, by going to other companies and licensing in their drugs so that we can develop them in the areas that we see as important. And then all the rest of them are foundations and patient advocacy groups and things like that. So that, again, we're missing the FDA and all kinds of other partnerships, but the point is it's a community effort. Now, when you look into the science, I don't want to go into the science too much, but again, there is a lot of research that's going on out there in the world. I don't know how many publications. If you look at the big companies, Roche, companies on that order, they spend $10 billion a year on research, right? So there's a lot of stuff out there you can look at and say, hey, what's similar? And this, we're talking about urgency and trying to find something quickly, trying to find a path quickly. So you don't have to do everything yourself. And you don't have time to do everything yourself. So just a couple of things to illustrate that this is a family map of the KIF proteins. There's 14 different families of KIF proteins. These all have similar functions in different contexts, and they're going to have things that are conserved. And there's a lot that we can learn from what other people have figured out. We can try to figure out how to apply that to our use. Similarly, if you look at the protein level, right, we talked about the feet that walk, right? So this represents KIF1A basically in the kinesin family. And this is moving the synaptic vesicles from the neuronal cell body out to the synapses, bringing all the machinery out to where the business end of the molecule is. So it's really important that these feet are working and the cargo carriers are working. But you can see this protein thymine that brings stuff back the other direction looks very similar, right? So maybe somebody's done some research on that. And similarly, myosin involved in muscles looks very similar. It has they do different things, but there are some similarities, and hopefully we can learn from those as well. And not only can we learn from the science, there's a lot of research. People have been testing billions of molecules in different assays, so we can take a look at the literature and see what aspects of that. We want to be very smart in how we do things, but also allow for some serendipity. Try to figure out other things that we can maybe take a shortcut or find an accelerated path to something that will be helpful. Then another layer deep, going into looking at the protein structure. And I won't geek out and go too deep into this, but I just wanted to highlight, you get in and this was really interesting for me as I was trying to get up to speed and understand what's going on. Here we talked about the gasoline in the car, ATP, right? This is the foot, this is the motor domain in a state, it's been frozen, and the structure like a camera, like an X-ray, or an X-ray of your bone, was taken when it had ATP in the binding spot, before the gasoline was used. So the gas is in the tank, it's not used yet. And then the same picture, X-ray picture, when the gas was used, right? And you can see it doesn't look a lot different here, but you can see here there's a bunch of movement, and it looks very different than here. So you can see that you have a kind of a Ziploc effect where this whole piece of the protein now has come up, and it looks very different, right? And when you look at the mutations all look like mutations that would interfere with protein-protein interaction. So how the amino acids of the proteins come together or don't come together. So I found that really interesting. And then you have people who've studied things and they've said, okay, here's wild type is when you have the normal tip formulated protein. And then when you have mutations, you can measure it. See, you put it in a little race, right? So this is velocity. How fast is it going down the track in this little test that they made, like a Pinewood Derby? So some of these mutations are the same or maybe even a little better than the normal one. And some of them are severely impacted or don't move at all, right? So this is kind of another aspect of what Dr. Chung was talking about yesterday, or even today, on kind of understanding some of the mutations and what their impact is, or what their role is. So, when you think through these things, then you can kind of say, okay, I understand a little bit about the gene and how the gene is expressed. I understand a little bit about the protein and some of the different, we heard yesterday about how the proteins walk along the microtubule. You put all of this information together and say, okay, now there are a few different things we can think about for how to change the dynamics in our children. And so it's very simply, there are a couple of categories, right? You can try to replace the gene. You want to maybe replace the bad gene that's there. Or maybe try to knock down that gene. Or maybe you can try to play around with some of the interactions and try to just give it a push so the KIP1A goes down the track. Or maybe there are things that are out there that have been working in other systems. We can try those, and I know those are some things that the team here has been looking at as well. So these are all different ways to think about how can we affect a positive outcome. And then this is just another way to represent it, right? And in a way, it's kind of, we don't know which one of these things is going to work, so let's try a few of them. And so you've got a funnel. I guess technically it should be going top down, but you get the idea. And so we've heard people talk about these. So small molecule, I showed a picture of that earlier. We heard about gene therapy and ASOs, antisense oligonucleotides, microRNA is another method of managing how genes are expressed. And then you want to put them in the cell line. So and the mouse models and see, are you seeing the effect that you want to see? And then you want to test to make sure that it's safe. And another way to look at that is, from one of the things that the biotech companies bring and the pharma companies bring is the process, is the scale, is the industrialized way of looking at these things, which sometimes is good and sometimes is too slow. But this is kind of a very simple way of looking at it, right? So there are things preclinical. Preclinical is obviously before you put things into people and into the clinic. And I highlighted in place or standard, right? So sequencing genomes, incredibly important. And that's, it's an amazing amount of work that this group has done to sequence genomes and understand them. The cell lines, having cell lines in place. It's amazing how quickly that was done. Mouse model, same thing. So you have all of the tools in place from a preclinical perspective. There are things you can add to that. There are things you can look for collaborations and other sources where you can make this a little bit more robust. But fundamentally, these things are in place already. You guys have done this already. And then safety screens. This is kind of from a biotech company or pharma company. This is standard. This is the stuff that they do in their sleep, right? So you don't need to build anything for that. When you get to non-clinical is just the other stuff. You're going to make sure that what you put into people is not going to be harmful, right? So you want to do build on the safety screens and go into we call it IND enabling. So investigation on new drug is what you get from the FDA that gives you clearance to test these treatments in people. So there's a certain package you need to bring together to be able to do that, data and information package. These are things that companies like this, they have. When you file a document, it's like thousands of pages, it's unbelievable. But you have people who know how to do that. It's not groundbreaking, it's not earth-shattering, you need to do it. Oops. And then you've got natural history, which was talked a lot about here, and it's so essential, so important to make sure that we collectively can look at treatments and find things that make sense or not, and actually are meaningful and not harmful. And then, maybe skipping down, the CMC is, sorry for the acronym, it's chemistry, manufacturing, and controls. And this is stuff for manufacturing with organizations all over the place. It's just standard off-the-shelf stuff. And then the compassionate use and the rare model, this is where coming together and working with the community. I know Dr. Chung has a whole plan and outline of how to do this. It's a way to do it urgently. It's a way to do it differently than if you're in a big company and doing this in diabetes or cardiovascular type indications. There's a new way to do things, and it's really by working together that we have the opportunity to do that. So that's kind of one thing I wanted to highlight. I think the other thing that's worth really highlighting on here, the team here, this organization, you have really done an unbelievable job, I think, leading the edge in how treatments are going to come about efficiently and effectively. And normally, when you go to a biotech company, they spend a lot of time putting this in place, everything green. But you have it, you've done it, right? And this is one of the philosophies that Luke talks about, that this community really is driving, is this idea of make it extremely easy for the biotech company, the pharma company to say, oh, this is compelling, this is great. Why wouldn't we do it? Let's do it, right? And so that's a lot of what we spend time talking about as well is, there is, at a certain point, like Luke mentioned, you're going to need to work with a company in order to really do the trials, or at least that's the hypothesis today. But by doing this, you can look and say, what you should never do is you have to make sure you're not totally dependent on that company. So you're not driven by their timelines, politics, fundraising, all that kind of stuff. By setting up like this, you have a lot more control in how you do it, and you're not hung up on artificial stuff. And that, I think, is something that's going to be transformative. It's not completely there yet, but there are a lot of activities ongoing. But I think as we progress here, if we continue with the urgency, I think it's going to be impactful. So, again, urgent collaboration is really about the community coming together, all of the stakeholders coming together and urgently doing what they can, asking questions. I have to tell you, working in a company where you've got the father of a daughter with a genetic disorder, conversations like you've never had before. You filter out the garbage so fast. Why are we talking about this? You're right. Okay, let's move on. What matters? Right? So anyway, that's all I have for now. Happy to answer questions or take them offline. Thanks.